Hi there. Let's look at glycolysis. So the glucose is taken in and two pyruvates are produced, and also 2 ATP and 2 NAD plus is taken in, and 4 ATP and 2 NADHs produced, right? So those are byproducts, and 4 ATP is the major product, right? Now, so perhaps you don't need the structures, and look at how to learn by mostly naming and enzyme naming first, and then later to the structures, right? Now, the cellular respiration has two locations, cytoplasm and the mitochondria. The cytoplasm exclusively for glycolysis pathway. It's a pathway, and Krebs is a cycle happening inside the mitochondria, right? So the glycolysis can happen without oxygen, which is fermentation, that is lactic acid and ethanol fermentation, right? If you do have oxygen, it's going to go into the mitochondria and it's going to do the oxidative phosphorylation. We call the OXPO. Okay, now, so that just to know where we are, right? And so where it is happening and where it's going. Now, the overall reaction of glycolysis has glucose 6 carbons breaking into pyruvate the 3 carbon units, 2 of them, and there are many steps, but in the process we're going to consume 2 NAD pluses and you're going to consume 2 ATP and you're going to produce 4 ATP, right? So the NAD plus is to pull out the hydrogen, so that means oxidation taking place, right? Now, the other thing, it's good to look at what is input, what is output, what is net. So the input is glucose is the substrate, the reactant, and also the 2 ATP and 2 NAD pluses, right? And output is the 2 pyruvates or pyruvic acids and the 4 ATP produced and 2 NADH produced, right? What's the net gain? 2 pyruvic acids per 1 glucose and 2 ATPs because you consume 2, produced 4, right? So that's how kind of you want to compare and write it in a simple format. Now, the other thing is, like I mentioned, there are 10 steps, but 1, 3, and 10th, those steps are the most important ones. They are no return, they are regulatory steps, they are big energy change steps. Now let's look at the glucose. So if you don't need the structure, just go with only with the names, right? But if you're aware of the structures, it's actually very good to understand. So the glucose is in the blood, in the water, the cells can take it, right? The hexokinase is basically for 6 carbons and it put a phosphate group from the ATP. Now the ATP put out of the 3 phosphate groups it has, puts the phosphate onto the 6th oxygen, right? So that's why it's going to be, that's the phosphate group we took from the ATP. So ATP become ADP, diphosphate, right? And then this one should be called glucose 6 phosphate or it should be G6P, right? So that's how the short name. And this step is a big step, like I said, first step, right? Because it uses energy. Okay. Now, so the next step is an isomerase. That means it's make an isomer from glucose to the fructose because it's the same number of carbons, hydrogens, oxygens, right? It's an isomer. That's why it's an isomerase. So this is fructose 6 phosphate then. You start with the glucose, you switch around 1 carbons, oxygens, and then you become fructose 6 phosphate, right? Or F6P, right? Now the next step is a big step. It's the third step. I have big arrows for them, and because it consumes ATP. Anytime production of ATP or consumption will be big steps, no returns, right? Now, here the phosphate groups goes on to the first, that one, first carbon hydroxides group, right? So that's the second phosphate came. Now what happened? The the fructose is imbalanced with the big phosphate groups hanging, so it's going to be breaking into 2 pieces, right? So fructose 1,6 bisphosphate, that's the purpose of that one, to break the molecule into 2, 3 carbon units. So bis is because the phosphate groups are in the same molecule in two different locations. It's not di, but it's bisphosphate, because di is like when they're interconnected, right? Now here, the purpose is 3 carbon units are actually different. If you don't need to put the numbers, but the dihydroxyacetone phosphate is 1. That's the right side, and the left side becomes um, glyceraldehyde 3 phosphate, right? Now, so let's yes, just to get the enzymes and the numbers correct here. Yes, that's the kinase. Kinase is putting on a phosphate group, right? And here, this is an aldolase. It breaks the 2 carbons into 2 different things, right? And not only that, between the DHAP and the G3P, what happens is fifth step is again an isomerase. There's 3 carbon units, there's changes happening in the second carbon, and that's why it's an isomerase. It's a simple step, it's an equilibrium, so no big energy change in this fifth step, right? Now G3P is the one moving forward to the up to the 10th step, not the DHAP. DHAP goes back to G3P. Okay, now, so the 2 NAD pluses are consumed actually for the 3 carbons, but just 1 NAD for each, right? So, and also triose phosphate dehydrogenase, that means you're removing the hydrogens, use NAD plus to take that hydrogens out. And so you have, now you have actually glyceraldehyde becomes glycerate or glyceric acid. This is a glyceric acid, or you can say um, yeah, glycerate, right? So this is also bisphosphoglycerate because there are 2 phosphate groups, 1 and 3, right? Now you notice the hydrogen is taken into NAD and it becomes NADH. The 2 just because there are 2 units, right? Okay. So if you can notice those, because the multiple choice questions are who is the reducer and what happened, oxidation, and so on, right? So that's why I'm trying to show that. Now, the 7th step is a big step again. So there should be some involvement of energy, right? Look at this one here. So the ATP is produced. Why? Because 1,3-BPG has extra phosphate and it is a high energy. And there's 2 of them. It's going to produce 2 ATP, right? Now you took, well, it's going to use 2 ADP, going to make 2 ATP. You left over with 1 phosphate, right? So this is happening with the um, yes, phosphoglycerate kinase enzyme and also the substrate level phosphorylation. This is called the substrate level phosphorylation. It's really kind of free money and free support you got, and that's why it's important, right? Now we end up with the 3-phosphoglycerate, and because it's a third carbon has a phosphate group, right? Now it's gonna swap. That's a mutase or mutation on the molecule, right? And it should be a simple step actually, and the phosphate jumps from third to the second, so now it becomes 2-phosphoglycerate, correct? So 2PG, right? So now, so we are here to move to the enolase, which is an important step. Why? Because if you remember, or if you've done the enolase production, there is water removal. Okay, and so in the enolase, what happens is you en for the double bond, O for the olate. See the double bond between the second and the third carbon. That's a double bond, right? So the double bond is, that's the ene, and then phosphate groups stay the same, right? And also this is a high energy molecule. This is the phosphoenol pyruvate. You should know what it is, and then it's called the PEP, and it's a high energy molecule and it produces energy. Okay, so how do we see that? The PEP can be, because it's high energy, it's going to dissipate the energy or put the energy into the, yes, it's 8, let's see, the 8 9, yes, 10th step, that's correct. And so in the 10th step you produce ATP from each PEP, phosphoenol pyruvate. There's 2 of them, that's why 2 ATP, right? So the 10th step gonna end up with the pyruvate, So if you have the H on the O negative, that's pyruvic acid, but normally under cellular conditions, it's just the pyruvate. So you're going to make two pyruvate from one glucose here, right? So 10 steps. Look at this one. Pay attention to a little bit of the enzymes. So 6 carbon uses hexokinase, and it's a big energy change step, delta G15. And then the isomerase is a simple one, change the glucose to fructose. And then the third step is important because there's an ATP or phosphorylation happens onto the first carbon, and it's a big minus delta G, about 15 kilojoules, right? So those are the key steps. So I put a different color so you can notice key steps only, right? And then aldolase is important. Why? Because you're producing two bases from the 6 carbon, the dihydroxyacetone phosphate, you call the DHAP. This is the acetone actually. Look at the acetone. You have to know the acetone at some point. So the acetone has one hydroxide group on the top, one in the bottom. That's why it's dihydroxyacetone phosphate, right? Now the other one, the seventh step, also important, the substrate level phosphorylation. So the phosphate came and ATP is produced. So that's why the seventh step is important. Next one. So basically I said 1, 3, 10, but 1, 3, 7 and 10 is important actually, right? Now this step is important, is the removal of the water in the enolase production. So that because of to make the double bond, right? And then the PEP is a high energy molecule, and then it produces ATP, and that's the big energy change. Now the fate of the pyruvate is plus in the presence of oxygen, oxidative phosphorylation, in the absence of oxygen, or goes to fermentation of ethanol or lactic, right? So that's the general idea. So try to write it on a paper 2-3 times and then match with the big fancy diagrams from the textbook or from the lecture slides, right? So writing on a piece of paper would make you understand the 10 steps and also the 4 important steps in the middle, right? Now the other way, by the side, so you need a long paper to put all these 10 steps, and by the side you put this energy diagram. So this one is actually Gibbs free energy, or you can also say potential energy. And what I'm showing, the first step is minus 15 kilojoules, and third step is minus 15 kilojoules, and the 10th step is minus 30 kilojoules, right? And why these are important? Because these are big energy gaps. These are regulatory steps of the glycolysis. So that means you have to know the enzymes, you have to know how much, why it's important, what's the substrate, what you end up with, right? Because those are the three most important steps. Now, so also the location of the process, the kind of to pay attention now, each location, basically glycolysis happens in the cytoplasm, right? And so the other, the no return steps, like I mentioned, either use the graph or use these steps and have a big arrow separate them. And every time you see the 10 step, you should pay attention to those 1, 3, and 10, right? And if you're aware of the energy, the kilojoule, that's because of the number of moles involved, right? Because the concentration levels are different based on the Le Chatelier principle. That's why we will have 15, 20, 30, 34, 39 different kilojoule values, right? Okay. So I think I mentioned about the enzymes, and let's look at the where we're going after this one. Yes. Um, yes. So once you have it, you also want to pay attention to what's the new. So after this, so notice the enzymes, notice the key steps, notice the energy amount, notice the graph, right? So once you're aware of this one, now what you want to do is you want to actually pay attention where you go after this one. So that is here. So look at the glucose 6 phosphate is the most important one. Glucose becomes that one. And this glucose 6 phosphate is actually connected to glycogen production, gluconeogenesis, and also pentose phosphate pathway. So be aware where you're going. Alrighty, thank you.